Hello, good morning. Well, it's morning for me. Welcome to another BIOL3465 Tropical Forest Ecology and Use lecture. This one is on biodiversity and floristics of tropical forests. So we've talked about some of the structure of tropical forests, how all those trees pack into an area to form this wonderful ecosystem, which is closed canopy, dark, humid, and so on. Now I want to talk a little bit about what are the species, what sort of, how the differing species make up a tropical forest. So what I want to talk about is biodiversity and how biodiversity changes at different scales in tropical forests. Now it's important to know this. It's important to know what sort of species make up a tropical forest. It's important to know how those species often vary within a tropical forest, for management, for conservation, for timber production, and so on. Because the floristic characteristics or the biodiversity characteristics of tropical forests will mean that, say for instance, timber production, you need to be able to understand where you're going to find those trees which you can harvest. It also has a big impact on the type of harvesting which can be carried out in a tropical forest, because obviously when you have a timber production system, not all species are useful for timber production. So what do? Do you chop everything down? No, it seems to be a waste of effort because there may only be one or two species. Okay, so I want to describe how species are different across a tropical forest. Okay, so I want to talk about how species are different at different scales. So diversity is mainly about species richness. Species diversity and composition can be thought of at different scales and can vary differently at different scales, as we shall see. Alpha or plot scale diversity is what is most often measured in a tropical rainforest. So alpha or plot scale diversity is basically the number of species you have as they vary over tens of or hundreds of meters. So most people would go and measure a plot in a tropical rainforest to try and get an idea of the sort of species which are in that tropical rainforest. Either that, or they would measure a transect to do their inventory of the species in a tropical rainforest to get some idea of, say, the density of different species. Beta or landscape scale diversity is the difference in species composition in different parts of the landscape, across the landscape. And this is over the scale of maybe hundreds or tens to hundreds of kilometers. So we're outside the variation in species across, just from a valley to a ridge, for instance. We're more looking at the difference in species, say from the east coast of Trinidad 50 kilometers to the west coast of Trinidad, or the difference in species composition between the northern range and the Paria Peninsula in Venezuela. That is beta or landscape scale diversity. Okay? And finally, gamma diversity is the total regional diversity that you find. So gamma diversity would be the number of species you find, say, in northern South America, or the number of species that you find, say, in the Amazon basin, or the number of species you find in Central America. And we've got a little graphic here which illustrates this. So alpha species diversity would be the number of species on a very local scale, on over the course of tens to hundreds of meters. So you would have different species within your plot, and your plot may stretch from a valley to a ridge, and it would have different species within 100 meters or so of each other, okay, or up to a kilometer of each other. And when you go to another part, say from the eastern northern range to the western northern range, you may find that you actually get different species, and that may be due to different reasons. Maybe due to rainfall gradients, probably is in Trinidad, or it may be due to historical biogeographic effects, so there were different refugia back in glacial times or something like that. But the difference that you see between these two plots at different in different areas is known as the beta species diversity. Okay? Now, beta species diversity will be low if the species that you find in the plot, this plot, is the same as the species that you find in this plot. Okay? So if the species are all the same, then the beta diversity is low. But if you go from one plot to the next plot, and at least some of the species, or the majority of the species, are different, then the beta species diversity will be high. So in Trinidad, beta species diversity tends to be quite high, mainly because of rainfall gradients. So if you do a plot in Matura, you probably get more forest, you would get some species were more adapted to higher rainfall. And then if you do that same, a similar sort of plot in, say, Chaguaramas, maybe down the island, you will find a completely different set of species. So the beta diversity will be quite high between them. Gamma species diversity is on a larger scale again, and it can be difficult to decide where to put your boundaries, but generally gamma species diversity is thought to be over thousands of kilometers, or hundreds, more like thousands of kilometers, and we would represent an entire region like Central America, or Northern South America, or the Amazon basin. Okay, so there's about 5,000 species in the Amazon basin, for instance. There's also about 5,000 species in Central America, and maybe we can class Central America with Northern South America. And when I say Northern South America, I'm talking about like all the forests which are west of the Andes, or the tropical forests anyway, which are west of the Andes or north of the Andes, and all the tropical forests of the coast ranges of Venezuela, all the way through to Trinidad. So about 5,000 species in the Amazon basin, about 5,000 species in the Central America / Northern America, and we can also count the islands of the Caribbean as a different region with their own gamma and gamma diversity. Okay, so gamma diversity is the total regional diversity, and I'll talk a little bit more about that later. Okay. Alpha diversity, as I've talked about already, can be important in management of forests. You want to know whether or how you can harvest, whether your target species are going to be in a low diversity area and have lots and have a high density, or whether it's going to be a high diversity area and have lots of your target species is dispersed amongst many other species. Beta species diversity may be important too, because if your target species is only found in one area of the country, that's the only area that you're likely to be interested in harvesting, and the other area isn't so much of interest. And gamma diversity may also influence your planning of your harvesting regime, whether you want to go and target this country or not, and so on. Okay, so diversity varies at different scales and can be important for management at different scales. So let's focus in on plot scale, or alpha species variation. That's the variation at scale of tens of meters. And most studies of species diversity, at least initially, were conducted on this scale. So when people first realized that tropical rainforests were really diverse, then when they wanted to characterize this diversity, or the difference between a tropical rainforest and a rainforest up in temperate regions, they studied the forests at this particular alpha diversity scale. And another reason why humans would tend to focus at this scale is because it's the scale which they can conceive most easily, because you can cover the alpha diversity area over tens or hundreds of meters in a human lifetime, and also in within the course of a field campaign. Maybe when you do your field work for a month or 2 months or so, this is the sort of scale that you're going to be working on. So when the scientists came down from the temperate regions and they were doing their surveys, they'd have a field season of a few months, this is the sort of scale that they were able to cover. So plot scale variation, or alpha species diversity, was the diversity which tended to be measured. So most studies are done on a hectare or quarter hectare plot in a tropical rainforest to find out the diversity of trees. Okay. Another reason why this sort of size or scale is used, because it is a traditional area which logistically can be done to inventory a forest for species, for understanding what species are production purposes. Recently when I say recently, over the past, 1980s, the past 30 years, some 25 to 50 hectare plots have been established. So most plots in rainforests, including in Trinidad, are established based on a hectare or quarter hectare plot. Some 25 to 50 hectare plots have also been established. Okay, so in Panama, on Barrow, Colorado Island, the first 50 hectare plot was established. And the reason why scientists were motivated to increase the size of the plots from 1 hectare to measure alpha species diversity is because the diversity of trees, at least in a tropical rainforest, was never completely captured in a hectare forest. So when sample area curves were done for 1 hectare plots, they still continued to climb after the while the after 1 hectare had been done. So that indicated there were many more species to be found even at the alpha scale, the alpha diversity scale. So 1 hectare plots, it was quickly realized, were simply not big enough to represent all the species which occurred in an area. So the Smithsonian Institution from America, from the United States, having enough resources and money, established a 50 hectare plot in Panama, which the US government controlled at the time. So that big investment in time and money and logistics could be protected. So this 50 hectare plot was established, and they certainly found an asymptote in the number of species per area, unit area, in this 50 hectare plot. Since the original 50 hectare plot was established, several more have been established around the world. The ones that I can remember are in Sabah in Malaysia. There was a 25 hectare plot, I believe, established in India and in a couple of other places around the world. And these plots are purely for scientific research, to try and understand how tropical rainforests are ecologically structured, and a large volume of tropical ecological research now is conducted in these 50 hectare plots. And the reason why these 50 hectare plots are so valuable to this sort of research is that you get enough replicates of species distributions and species reactions to abiotic changes across the landscape that you can statistically represent what's going on a local ecological scale, which influences the distribution and persistence of species. We don't have a 50 hectare plot in Trinidad, but we do have a lot of 1 hectare plots, and I'll show you the distribution of those in a minute. Within these plots, traditionally trees 10 centimeters dBH and above were measured. You know what dBH is? Very good. In these 50 hectare plots, however, trees were measured down to about less than 1 centimeter dBH, usually about 1 centimeter dBH. Now the reason why 10 centimeters dBH is used is quite often because in many forests mortality rates in small trees are much higher than mortality rates in large trees. And that is due to the fact that the larger trees have better access to light and resources because they're taller and they reach up into the canopy. The smaller trees will die more regularly and faster because they are still shaded out by the larger trees. And it's around 10 centimeters dBH. When a tree reaches about 10 centimeters dBH, the mortality rate is usually low enough that you can be fairly sure that this tree is going to survive into the future. Okay? So if you measure underneath 10 centimeters dBH, you're going to have to be prepared to remeasure and completely look at the re-inventory the plot every 2 or 3 years, because those stems are going to likely die and be replaced very quickly. In these 50 hectare plots, stems were measured down to 1 centimeter dBH, and that was important because scientists could then study recruitment and regeneration, which is a critical part of understanding the distribution of species. But it meant that these 50 hectare plots, because they were incredibly large, and we're talking about what, 1 kilometre by half a kilometre plots. All the stems down to 1 centimetre dBH measured. Okay, we were talking about, I think about 300,000 stems. And the initial setting up of this 50-hectare plot in Panama, I believe, took about 3 years. And after 3 years, they turned around and started re-inventorying the plot once again. The re-inventory of the plot, I think, took a quicker time because most of the trees were already tagged and the outlines of the plot were set out, and that took about a year. And this 50-hectare plot in Panama is re-inventoried every 3 years or so. So it's a lot of work, a lot of resources. They have several full-time people going in there and remeasuring all the time. Okay, it's mainly the trees which have been studied in a tropical rainforest, mainly in recognition that it is the trees which are really the structural components and also the most economic components in a tropical rainforest. Non-trees tend to be less well studied, and it's thought that the non-tree diversity is pretty much underpinned or reliant on the tree diversity. So it's really the tree diversity which tends to be studied, and non-tree diversity is less well studied. Obviously you will have scientists who are very interested in different taxa, so things like the birds, the butterflies are quite often well studied. In recent years, in the last 20 years, with the drive to understand biodiversity levels around the world, the insects have come into focus, and there's been a fair amount of work on trying to understand how many insects there are in these tropical forests. The types of sample units, however, which are used for non-trees are usually not plot sample units. And there are a plethora, many different types of sampling strategies used for different taxa. Too many that I'm not going to go into all of those now. So, what is the range of plot scale or alpha species variation in tropical forests? Well, generally the range is from approximately 40 species to over 300 species per hectare. Trees 10 centimeters dBH and above. These numbers of species will be higher if you measure down to 1 centimeter dBH. But the vast majority of plots which spread across the tropics tend to be about 1 hectare or even a quarter hectare. 1 hectare is 100 meters by 100 meters. A quarter hectare is 50 meters by 50 meters. And the trees are usually measured down to about 10 centimeters dBH. That is a de facto standard. It's logistically what is achievable within, as I said, a month or so. It's approximately 40 species to over 300 species. This 40 species tends to be found, say, in Central America. In Trinidad we would tend to find 40 to 50, maybe a few higher species per quarter hectare plot or 1 hectare plot. These high numbers of species tend to be found in the Western Amazon basin. Now, Western Amazon basin, as you may remember, was one of the few areas which remained under forest cover during the last glaciation. So that may explain why the species level diversity, the alpha species level diversity, is so incredibly high. So some images of plots. Remember this image from before? This is actually the image of a 1 hectare plot, okay, and the trees are scattered within it, okay. I can't remember, I think this plot had species richness of 40 species. Okay, this teak plot obviously had a species richness of 1 because it was a plantation. Most of these plots, or often in these plots, you would find that the trees are individually marked, and this is important if you're going to do any sort of dynamics in these plots. For timber production, trees can be individually marked and remeasured at different times, say every 3 years or so, to assess how quickly species grow. Now you can imagine that knowing how quickly species grow is going to allow foresters to understand how long they need to leave a forest before it accumulates enough timber to go in there and harvest. Okay, so plots can be very useful in timber inventory, so that you can remeasure and measure growth rates. And that is one of the main reasons why alpha scale diversity is measured across such large areas, because a lot of these plots are forestry inventory plots. And I've got a picture here of a plot. Now this is very unusual. This plot, I think it's about 1 hectare in size, or maybe 1/4 hectare in size, has been marked out in a primary forest. I think it's primary forest originally because these trees have no lower down branches, and it's been cleared around this plot. So this is an example of what a hectare or 1/4 hectare plot would look like if you isolated it, took it out of the forest. This is actually down in the Amazon basin near Manaus. You may have heard of this research project. This was the fragmentation research project, where fragments of forest of different sizes, ranging from 1/4 hectare, 1 hectare, 10 hectares, 50 hectares, 100 hectares, were all marked out, and the land was cleared around them. And the idea of this experiment was to see what sort of effect this fragmentation had on the remaining forests. This experiment was started about 25 years ago, and it's been running ever since, and they're just starting to bring in data now to find out what is happening to these remaining plots and the biodiversity within the plots, the tree biodiversity and the animal biodiversity. I think you can see that there's a lot of edge effects there going on around this plot, isn't there? There's another fragment in the distance over there. A massive project. Unfortunately, I think they're finding some useful data, like things like what happens to the bird populations in these fragments, what happens to the mammal populations, and insect and variation insect populations as well. So that's what a plot would look like. Okay. In Trinidad, there are a whole bunch of plots which have been established, and they're called permanent sample plots. In Trinidad, they were established mainly for assessing growth rate, particularly of timber trees in a tropical forest. So about 144 1 hectare plots were established by Forestry Division and measured at different times, and they're scattered across the island. The red ones were established by Forestry Division. The purple ones up here were established by UE around 2000, or between 2006 2007, and all the species were measured down to 10 centimeters dBH. In the forestry plot, only the timber species were measured down to 10 centimeters dBH. The Forestry Division plots were 1 hectare plots. The Matura in National Park environmentally sensitive plots were quarter hectare plots, and only 24 were measured, okay, to try and capture the alpha species diversity within the Matura National Park. And I've got some species lists there which you may not be able to read. This was a poster that Carlton Roberts and myself put together for presentation at a regional workshop up in Santo Domingo a couple of years ago. Okay. So the number of species range from about 40 up to about 300. 40 is a fairly characteristic, between 40 and 80 is a fairly characteristic number of species in Central America, Northern South America and the Caribbean. Over 300 species have been found in 1 hectare plots in the Western Amazon, as I said. Now we can contrast this with temperate species levels, and I think the maximum number of species which were found in the highest diversity temperate forests, which happen to be the broadleaf forests of the southeastern United States, and the maximum number of species there is about 10. So we're talking about almost an order of magnitude greater number of species per hectare in the plots of the tropics. Okay. So how does this species richness vary? It can vary quite well with seasonality and amount of rainfall, particularly in the neotropics. But there is some debate on whether this species richness is ecological of ecological origin or of historical biogeographical origin. So in other words, the processes which determine species richness, say the difference in species richness in a 1 hectare plot between the Amazon basin and Central America, Northern South America for instance, may not be completely due to the current seasonality and amount of rainfall, but may also be due to the disturbances, the historical disturbances of these forests during the last ice age, because Central America and Northern South America were much more impacted by drier conditions than the Western Amazon. And it just so happens that today the Western Amazon are one of the wetter parts of the neotropics, and Central and Northern South America are substantially drier and have a greater seasonality. Okay, so seasonal species richness tends to vary with seasonality and amount of rainfall. Okay, and we've talked about some of the processes. Well, we will talk about some of the processes later on about why this is. It's probably due to stability of the environment allowing specialization of species in particular niches and so on, and other reasons that we'll talk about in other lectures. Alright, so species composition varies across the plot scale. Quite often landscape position at the alpha species scale is going to vary a lot on landscape position and whether the area is very topographically varied or not. So ridges and valleys quite often will have different moisture characteristics, different nutrient characteristics, and therefore will support different species. Here in Trinidad, we class our forests or classify our forests as Crapo-Guatacare forests, at least the forests in the lowlands. Crapo-Guatacare forests. Those are the two main species which we tend to find, and they are actually separated into different landscape positions. The crapo trees, or the Carapaguayanensis trees, to give it its scientific name, tend to be found in the valleys. And the guatacare trees, or Eschwellera subglandulosa, are trees which tend to be found on the ridges. And that doesn't mean that they don't occur together, and they do. The forest that we went out to, the arena forest there, had both Eschwellera and Crapo in the area that we were working in. Okay. Yeah, so the species composition will vary across a plot scale and within an alpha scale on topographic basis due to different resource availabilities, soil, water, and soil depth, and so on. Okay, I think that makes sense, really, doesn't it? So we tend to have high species diversity in tropical rainforests, but we would also have high genus and family level diversity. So in a tropical rainforest at a plot scale, we would tend to find, say, maybe 40 or so species, 50 or so species in Trinidad. But the number of genera and families which those species are spread between is also very high. So we would quite often have, if we have 40 species, we may have something like 25 to 30 genera, and maybe even 10 to 15 or 10 to 20 families. So quite often a family is represented by a single genus, or a family may even be represented by a single species within our plot. Okay? So that means that there tends to be a lot of families represented in tropical rainforest plots at an alpha species level. If we contrast this with other parts of the world in the temperate regions, quite often the species diversity, the species are actually belong to all the same genus or all the same family. In Western Australia, where I grew up, the species diversity is some of the highest outside of the tropics. This is a Mediterranean ecosystem. They have these shrublands, and within 1 hectare of shrubland you may get 30 to 40 species of shrubs. Okay? The genus level diversity, however, is much lower. So those shrubs may belong to about 4 or 5 main genera. Okay? And this reflects the fact that there's been recent invasion and adaptive radiation of those genera in recent times. So the species level diversity tends to be high, but because tropical rainforests tend to be much more ancient and older, their genus level diversity tends to be high as well, and also the family level. Okay, so species level is high in tropical rainforests, but genera and family level diversity is also very high. Okay, let's move on up to the next scale of variation of species. That's the landscape or beta variation of species, and that's where we're looking at the scale of hundreds of kilometers. Okay, so tree species vary more across 100-kilometer scales than in temperate regions. So generally in tropical regions we will find that two plots or sample areas separated by about 100 kilometers or so will have different species, even though the ecological conditions are pretty much the same. Same valley bottoms and ridge tops. in both places, but we would find different species in the valley bottoms and on the ridge tops in the different species. A good example would be that would be the species of trees that you find between the northern range of Trinidad and equivalent areas in the Paria Peninsula in Venezuela. So the eastern end of the Paria Peninsula is very similar in environment or habitat to the eastern end of the northern range. But the species that you find can be very different. The dominant species are actually very different between these two areas. There's a guapa genus which is simply just not present in Trinidad, but it is one of the dominant genera in Venezuela. Some of the species like Mora in the Leguminosae, which is fairly common and dominant in the eastern part of the northern range, is completely absent from the Paria Peninsula. There are some tree species, obviously, which are the same and are found in the northern range and in the Paria Peninsula, but generally the dominant species are quite different. And this is an example of landscape variation or beta scale diversity in species. Okay? The beta scale diversity actually varies quite widely across the different regions of the neotropics. Generally, species turnover across the landscape is high in Central America, okay? But you tend to have the same families and genera, okay? So there tends to be the same genera like ficus and so on. Same genera like ficus and so on. The families are all pretty much the same. The Annonaceae, the Leguminosae or Caesalpiniaceae and so on are pretty much the same across these different regions. Okay, they may be more or less important in different parts. So, for instance, the Myrtaceae family tends to be very important up in the Caribbean. The Papilionaceae family tends to be important in the Amazon. Those are the legumes. Okay, so different families, well, they all occur in the different regions, but some will be more dominant than others in the different regions. Generally, beta species diversity is very high in Central America, and as I said, we can class Northern South America in that bracket as well. So beta species diversity tends to be quite high, okay, and that includes Trinidad. And yes, we do have quite a high beta diversity in Trinidad. However, the areas which have the highest alpha species diversity in the Western Amazon actually have low levels or much lower levels of beta diversity in the Western Amazon. So the plots which you measure may have a lot of species in them, over 300 species in some cases, but if you go 100 kilometers in the other direction, you're going to find pretty much the same species in your sample area. So there tends to be fairly low beta diversity. Okay, so that means that there are about 5,000 species in the Western Amazon and about 5,000 species in Central America. Those 5,000 species in the Western Amazon tend to be spread throughout the Western Amazon. In Central America, on the other hand, those 5,000 species have a much more restricted distribution. Okay, why is this so? It's thought that it's mainly due to historical effects. So in the Central American landscape. Okay, the variation is due to different species being separated by topographic features. So if we look at the example of Trinidad and the Paria Peninsula, the Northern Range and the Paria Peninsula, they are separated by that big gap there, which is represented by the dragon's mouth. During the last ice age, when the sea level was lower, that would have all been dry forest. There would have been rainforest in the Northern Range and on the Paria Peninsula because they're higher due to orographic effects, and so they would still be pretty much what they are today, maybe more restricted in distribution, but still patches of rainforest. But they would have been separated. So this separation would allow different species to evolve, and so you would tend to find a much higher beta species diversity once the wet times come back and the forests tend to be more contiguous now. Okay? Lack of variation in Western Amazona may be due to a lack of topographic variation, but probably more importantly a lack of disturbance during glaciation. The most recent paradigm predicts that Western Amazon was wet enough during the last ice age to still support tropical rainforests, so it wasn't actually disturbed, so the species have no reason to have their dispersal restricted, and so they spread throughout the region. Okay? Variation on the scale of hundreds of kilometers probably reflects rainfall gradients as well. Yeah, so you're going to have some species which specialize in the drier end of the spectrum or the wetter end of the spectrum. So you're going to get changes in your species composition as you move over these hundreds of kilometers. And in Trinidad we see that gradient even over 50 or to 80 kilometers. We get pretty good variation, and that's on the boundary between alpha species diversity and beta species diversity. I think we can probably call it beta species diversity, and that tends to be high because of rainfall gradients. And this rainfall gradient going from the east coast inland is a fairly common one at this particular latitude on eastern sides of continents. You see the same steep rainfall gradient with rainforests on the coast grading into drier forests the further west you go. You see that same thing in Australia on the northeastern coast of Australia at around the same latitude, and it's due to the trade winds bringing in moisture and keeping those rainforests alive, but the rainfall not penetrating too far inland. Should have a diagram for that, shouldn't I? Sorry about that. Alright, let's just talk a little bit about, well, last slide, a little bit about regional variation or gamma species diversity. Okay, there are very few species common between the different tropical regions, very few species common. So the Neotropics has its own species, Africa has its own species, and Southeast Asia has its own species. Africa and Southeast Asia, because they have more consistently been joined by land, tend to have more species in common than the Neotropics and the other two. But that doesn't mean that the Neotropics don't have species in common with these other two, particularly Africa. There are some species, and it's thought that those species dispersed from Africa to the Neotropics subsequent to division of those two continents. Okay, some of those species would be like the yellow mang that you find in Trinidad, or Symphonia globulifera, to give it its scientific name. It's found here in Trinidad. It's also found in Africa. There's a genera which is common between a Trinidad species and an African species, and that would be the crapo, the carapa. There is a carapa species found in Africa, in West Africa. It's fairly common, and the crapo tree, the Carapa guianensis and the Carapa procera, are also found here in the neotropics. Again, it's thought that dispersal was the reason why we tend to find those genera both in Africa and in South America. But these common species are the exception more than the rule. Genera common tend to be more usual. So genera such as Ficus, for instance, is found not only in the neotropics but also in the African and Southeast Asian tropics as well. At the family level, families actually are shared between all these different tropical regions. So the same families that you find in the neotropics, Africa and Southeast Asia, you will find across all those different tropical regions. So the things like the Ananaceae, the Papilionaceae, the Gramineae, they're going to be found across all these different areas. Okay, so that indicates that some of these families arose before Pangaea broke up, or, and this is probably most likely, there's been enough time, they've been around long enough that they've been able to find roots at different periods in history where the continents have been joined by various means to be able to move between the different regions. So many of these families have been found to have first evolved after Pangaea split up, but because Pangaea had a link, say for instance South Australia had a link between Australia or the East Asian tropics through Asia and North America down into the neotropics at some stage, and all of that land route was covered in rainforest at some time. Those families have been able to exchange and disperse between the different regions. So families, because they are higher order taxa and they have been around for many millions of years, they tend to be found between the tropical regions. Okay, they have been around when dispersal between the regions is possible. So this commonality between families does reflect historical biogeography. But when it comes to gamma diversity between these different regions, it is very high, okay, at the species level. Not so high at the genus level, and very low at the family level. The gamma variation, say, between the Amazon basin and the Central America, again, tends to be quite high. Okay, there is a few common species between the regions, but there are also quite a few species which are unique to, say, the Amazon basin and they're not found in the Central American region. Okay, some of the more widespread species may be some of the wind dispersed species like this pui, yellow pui, okay, which can spread through the Amazon basin. The Maurice palms are also a group which has made it from the Amazon basin, where it's thought to have has its origins out into Trinidad, although humans have been implicated in that change. Okay, so that's the end of the lecture. What I want you to take away from this is that tropical forests are very diverse. Okay, species tend to occur at low densities, so there's a whole bunch of species in a small area, which means inevitably that each species will occur at fairly low densities, and that has a big impact on how you manage your tropical forests, whether your goal of management would be timber production all the way through to conservation. Okay, take a read of Gazul and Shiel, books available in the bookshop. They've got a good section on alpha species through to beta species, gamma species diversity on pages 155 to 155 to 155 to above, and you can also find information on the different rainforests through time and so on, from about 103 when they start talking about flowering plants through to through to 127, and they talk quite a lot about the differences in species, family and genera between the different regions around the world, neotropics, Africa and Southeast Asia. Well worth reading. Okay then, thank you. Bye bye.